आपके ब्रेड में लग जाता है, ब्रेड में, that rhizopus, that is a fungi. We know mushroom, we know this is agaricus, scientific name is agaricus, you know, most common mushroom that we eat. We know it is a fungi. It is a fungi. This is basidiomycetes. We know this is basically basidiomycetes. So yeast, we know this is ascomycetes. This is ascomycetes. Is this clear? If somebody has heard of slime molds, एक term होता है, sometimes we use this term slime molds. Have you heard of slime molds? So slime molds. We know slime molds are basically, they are studied under fungi. We know they are basically studied under fungi. ठीक है? But in fact, ये जो slime molds हैं, slime molds are cusp. They are basically cusp. Cusp मतलब बोले तो जैसे टोरियन और वर्गो के बीच में, ठीक है? It is a cusp. That means when I say slime molds are basically cusp, that means they have some properties. They have some properties of plants, and they have some properties of animals. They also have some properties of animals, you know. This is very clear. So slime molds are basically cusp, but we study them under fungi. इनको हम fungi mycologist, they basically study under fungi. लेकिन plants में भी now recently मैं आपको ये बता दूं कि slime molds, if you look at the animals perspective, slime molds are also regarded as protista. Slime molds are also regarded as protista. Is this very clear? So now they are also regarded as protista, matlab they are also studied by the animal people. The animal people also study them, slime molds. ठीक है? और किस क्लास में फॉल करते हैं, किस कैटेगरी में फॉल करते हैं? They fall under, we study them under either myxomycota. Normally we study them under myxomycota. ठीक है? So class is myxomycota. तो किसी ने क्लास में रख दिया है, किसी ने इसको सब डिवीजन में रख दिया है. It depends on the classification. According to Ainsworth classification, अगर हम Ainsworth की क्लासिफिकेशन देखें, if we look at the classification of Ainsworth, Ainsworth has kept it under a class. So we have class myxomycota. ठीक है? So different people have classified it differently. You know? Now the question is, see, when we talk about the slime molds, when we talk about the myxomycota, let me take a new page. When we talk about this myxomycota, ये जो myxomycota है, we know because they are basically studied by mycologists. I told you they are studied by people who study fungi. There is mycologist, but in fact it is kept under animals also. I just told you it is kept under animals. We classify under protista. The reason that it is studied by mycologists, what is the reason that it is mainly studied by mycologists? Because it is found under those environments where basically fungi grow. So in those climates, in those environments where fungi grow, myxomycota is also found. And that is why one of the reasons is that it is studied by the mycologist, because it is more encountered by the mycologist under those environments where fungi is basically grow. Okay. Now, when you look at this myxomycota में ये जो members of myxomycota, if I look at the members of myxomycota, we know these members have a vegetative phase. एक यहाँ पे vegetative या somatic phase है. We have a vegetative and somatic phase. And then we have a reproductive phase. We have a reproductive phase also. So we have a somatic or vegetative phase, and we have a reproductive phase. Now, if you look at the somatic and vegetative phase, if I look at the somatic phase, we know the somatic phase, this is very similar to amoeba. This is very similar to amoeba, you know. This is very clear. Or it is similar to plasmodium. It is very similar to amoeba. It is very similar to plasmodium. Matlab, it can move by phagocytosis. It shows movement. It shows locomotion. It basically shows locomotion. Locomotion by what? Locomotion by phagocytosis. It shows locomotion by phagocytosis, you know. Okay. So that means if you look at the vegetative structure of myxomycota, this is very similar to amoeba, or it is very similar to plasmodium. It is also very similar to plasmodium. Okay. When I say plasmodium, matlab ye jo vegetative structure hai, we know this vegetative structure under certain conditions, it becomes multinucleate. It can become multinucleate, and then we call it plasmodium. Is this very clear? So somatic structure is very similar to certain protozoas, it is very similar to certain protozoa. And that is why we can keep myxomycota or the members of myxomycota under the animals, we study them under animals. Now, if you look at the reproductive phase, we know in the reproductive phase, it is basically reproduces by means of spores. It basically reproduces by means of spores. And the spores are formed. These two spores, and they are basically formed on a fruiting body. They're basically formed on a fruiting body, We have a fruiting body also. And this fruiting body, this is called as sporophore. What do we call this fruitification of fruiting body as? Sporophore. So this sporophore is a spore-bearing structure. Okay. Now, when you look at these spores, if I look at this spore, I say this is spore. Now, what is amazing here, that these spores have cell wall. These are spores and they have cell wall. This is a cell wall. भैया animal में तो cell wall होती नहीं है. Animals never have cell wall. Cell walls are either present in plants. Even fungi have cell wall. Fungi also has cell wall. So if you look at the reproductive structure, reproductive structure is very similar to what? This is very similar to fungi, or this is very similar to plants. This is a plant character, or this is a fungal character. Is this very clear? So that is why I'm saying कि जो members of myxomycota है, जो members of myxomycota are basically cusp. They are basically cusp. They have certain features of fungi, they have certain features of plants, they have certain features of the animals. Okay. Even if you look at the somatic cells, if I look at the somatic phase, I told you somatic phase is very similar to animals. Somatic phase is very similar to animals. Not only in terms of locomotion, it is very similar to animals. But even if you look at the nutrition, अगर मैं इनकी nutrition की बात करूं, so nutrition of this myxomycota, the nutrition of this myxomycota is also very similar to amoeba. It is very similar to amoeba. मतलब जैसे it engulfs, जैसे होता है ना amoeba will engulf the certain maybe certain bacteria or something. Okay, by phagocytosis it will engulf some certain extracellular material. Amoeba में क्या होता है? अगर मैंने कहा amoeba है, ये amoeba है, तो यहाँ पे we have extracellular material. This extracellular material will be engulfed by the cell via phagocytosis. We have a phagosome. The phagosome goes and fuses with the lysosome. याद है ना? The phagosome goes and fuses with the lysosome. So all the content to be digested will ultimately go to lysosomes. मतलब ये जो digestion है, we know digestion in amoeba is what? This is intracellular. Digestion क्या है यहाँ पे? Intracellular. This is intracellular. ठीक है? तो animals से हम amoeba की बात करें, हम paramecium की बात करें, digestion we know is basically intracellular. ठीक है? तो अगर हम myxomycota की बात करें, myxomycota में भी the digestion is very similar to amoeba, that is intracellular. In members of myxomycota, the nutrition is intracellular. ठीक है? लेकिन in most fungi, अगर हम fungi की बात करें, in most fungi we know the nutrition is extracellular. The nutrition is extracellular. First, the fungi will secrete certain enzymes. The enzymes will digest this digestive material is digested outside. It is broken down outside the cell. तो fungi क्या करते हैं? They will release certain enzymes which will degrade the food material. And once the food material is broken down, it is then taken up by the cell. So this is extracellular degradation. Most of the fungi we know they show extracellular degradation. लेकिन अगर हम myxomycota की बात करें, अगर हम amoeba की बात करें, वहां पे the digestion is intracellular. So there are vital differences, Somatic cells are very similar to animals. लेकिन अगर हम reproductive structures की बात करें, reproductive structures are very similar to plants as well as the fungi. And that is why I am saying कि जो members of myxomycota है, it is a cusp. It is a cusp. मतलब plants के भी characters हैं, animals के भी characters हैं, कुछ fungal characters भी हैं यहाँ पे, ठीक है? And that is why this becomes so interesting, And these members of myxomycota are also called as slime molds. इनको हम क्या कहते हैं? We call them as slime molds. Okay. So this is very clear. मेरे मन में आया तो मैंने आपको बता दिया, बिकॉज दिस इज इंपॉर्टेंट। ठीक है, सो वी आर स्टार्टिंग विद फंजाई, we are starting with fungi, and before we start with fungi, we know who's the father of Indian mycology, who's the father, and write sab kuch, who's the father of Indian mycology. We know this
हाँ,
is E.J. Butler. The name is, this is E.J. Butler. ठीक है, he is the father of Indian mycology. This is Edwin John Butler. अगर full form याद नहीं रखनी है आपको, E मतलब Edwin. This is Edwin John Butler. ठीक है, so he is the father of Indian mycology. And now, who's the founder of mycology? Or sometimes we say, who's the father of mycology? ये तो Indian है ना, but actually who's the father or who's the founder of mycology? Founder or father of mycology? We know this is Antonio Myceli. Who's this? Antonio Myceli. He is said to be the founder or father of mycology. And he also have written a book. He has also written a book. He has also authored a book which is called as Nova Plantarum Genera. So आपको लिखना नहीं है, लेकिन this is Nova Plantarum Genera. So this is the book which is written by Mr. Antonio Myceli. Okay. So apart from this, when we are talking about fungi, see, when we talk about fungi, भाई fungi में कुछ थैलस भी होगा, मतलब कुछ एक बॉडी भी होगी ना, एक सोमेटिक बॉडी भी होगी. जो सोमेटिक बॉडी है, ये जो सोमेटिक बॉडी है, सोमेटिक बॉडी, और समटाइम्स कॉल इट प्लांट बॉडी. प्लांट तो वैसे टर्म नहीं यूज करना चाहिए, वी शुड नॉट बी यूजिंग दिस टर्म प्लांट, बट समटाइम्स बोल भी देते हैं कि प्लांट बॉडी या सोमेटिक बॉडी. ठीक है, दिस इज अ थैलस. वी नो दिस इज बेसिकली अ थैलस. तो जो सोमेटिक बॉडी है, वी कॉल इट अ थैलस. एंड वी नो दिस थैलस इज मेड अप ऑफ वेरी थिन थ्रेड लाइक स्ट्रक्चर्स. जो थैलस होता है, जो इसकी बॉडी है फंजाई की, दिस बॉडी ऑफ फंजाई which is a thallus is made of very thin thread like structures, thin thread like structures, and we call it what? We call it hyphae. We call these thin thread like structures as hyphae. ठीक है, and when we have multiple hyphae, ये जो multiple hyphae हैं, ये देखो, normally what happens, it's not a single hyphae, but these multiple hyphae are present together. These multiple hyphae, we know they are present together, and then they form a kind of a thallus. When multiple hyphae, we have multiple hyphae are present together, okay, and they form a structure. They form a structure which is called as mycelium. So many hyphae, when forming a network, they are forming a network. They're present together. Network, they're present together. They form a structure called as mycelium. So what is a thallus? A thallus is nothing else. It is basically a mycelium. A thallus is primarily this is a mycelium, and sometimes this mycelium, it gets organized into different kinds of structures. We know mycelium can get organized. This can get organized into different types of structures. Different type of structures. For example, we have rhizomorph, we have plectenchyma, we have pseudoparenchyma. So we have sclerotium. What are all these? Plectenchyma, pseudoparenchyma, rhizomorph, sclerotium. What is all this? These are nothing else. These are different kinds of structures which are basically formed by mycelium. This is very clear. So matlab mycelium can also form a certain degree of tissues. They can form certain degree of tissues also. So certain degree of tissue formation is also seen under certain conditions. And these tissues which are formed, we have different terms. These tissues, like structures which get formed by this mycelium, we give them different names. They are given different names. So just for your knowledge. So now we know what is a hypha, what is a mycelium. The fungal body is called as a thallus. Now, how do I define fungi? If I have to give you a definition of fungi, we know fungi, they are eukaryotic. पहली चीज तो याद रखनी है. All the fungi are basically eukaryotic. They are spore bearing. they are spore bearing, they are achlorophyllous, chlorophyll नहीं होता है इनमें, तो they are non photosynthetic, so they are achlorophyllous, ठीक है, organisms, so they are achlorophyllous organisms, ठीक है, that reproduce asexually as well as sexually. Very simple, very simple, ठीक है. And we know इनके जो cells होते हैं, the fungal cells, they also contain a cell wall. Fungal cells also contain a cell wall, so they have a cell wall, and the cell wall, we know the cell wall either contains chitin, we know their cell walls have chitin, or sometimes their cell walls, मैं cells की बात नहीं कर रहा हूँ, मैं walls की बात कर रहा हूँ, cell walls, their cell walls have chitin, their cell walls may also contain cellulose, their cell walls may contain mannan, their cell walls may contain glucans, ठीक है. So their cell walls have cellulose, chitin, mannan, glucans, and other different kinds of polysaccharides can also be present, different components can be present in the cell walls, ठीक है. Now when we look at the hyphae, look at this, अगर हम hyphae की बात करें, I am telling this is the primary structure is hyphae, सब कुछ यहीं से start हो रहा है, everything starts from a vegetative structure. We know this vegetative structure, this is called as hyphae, and this hyphae, let me tell you, either it can be a single, ये देख रहे हो, the entire hyphae is a single cell, देख रहे हो, it is a single structure, not divided into cells. Sometimes we have hyphaes which are not divided into different cells, you cannot see any division, it is a continuous structure, you can see it is continuous, देख रहे हो, पूरा continuous है ये, cytoplasm is present throughout, this is a continuous structure, hyphae, with no divisions, no septa. Divisions are called as what? ये जो divisions होते हैं, जो compartments होते हैं, so compartments are formed by means of septa, structures called as septa. So there are no septas over here, it is a continuous structure, and we see this continuous structure can have multiple nuclei, we can have many nuclei, so it is multinucleate, it can be multinucleate, and we call this multinucleate structure. Let me tell you, this multinucleate structure, this is called a cenocytic, this is a cenocytic structure. So when I say multinucleate, when I say cenocytic, मतलब क्या? Cenocytic hypha, अगर मैं कहूँ कि cenocytic hypha मतलब क्या? It is a multinucleate structure without any wall, without any divisions, without any compartmentalization, without any division. So this is a cenocytic structure. But sometimes we see, sometimes we have the compartmentalization. जैसे मैंने आपसे कहा, sometimes we have the compartments which are formed, ठीक है. मतलब ये जो hypha है, hypha can be divided by means of walls. This hypha can be divided by means of walls, you know, cross walls, we call them as cross walls. Cross walls can divide this hypha into different cells. Now we have different cells, ये एक cell है, ये हमारी दूसरी cell है, so it becomes a multicellular structure, it can become a multicellular structure. और ये जो cross walls हैं, we know these cross walls are called as septa, we know these cross walls are called as septa, ठीक है. So this should be very clear, and we know ये जो fungal cells, now look at the individual fungal cells, we know जो fungal cells हैं, they can be uninucleate, they can be binucleate, they can be multinucleate. So we have all different kinds of conditions, you know. Fungal cell, if I talk about a fungal cell, we know fungal cell can be cenocytic. Cenocytic मतलब कोई division ही नहीं है, कोई cross walls नहीं है. For examples, if I give you example is we have oomycota, oomycetes. ये जो oomycetes हैं, this is one of the categories within fungi. Oomycota, oomycetes, सबने नाम सुना होगा oomycetes का. तो oomycetes का जो thallus होता है, if you look at the thallus, in this thallus ये जो hyphae होते हैं, the hyphae we know are cenocytic and there is no septa. It is a continuous structure, it is a multinucleate structure, and it is aseptate. मतलब septa नहीं है तो मतलब aseptate. This is a aseptic structure. Example is oomycetes. ठीक है, और मैंने अभी आपको कहा कि sometimes ये जो fungal cells हैं, the fungal cells can be uninucleate, they can be binucleate, they can be multinucleate cells. For example, we have ascomycetes, uninucleate. Binucleate, we have basidiomycetes. Multinucleate, we have deuteromycetes. Deuteromycetes. Look at all these examples over here. So we can have different kinds of cells, in fungi. Okay. Now look at the septa. I told you the cross walls are called as septa. So this is a hyphae. In the hyphae we have cross walls. The cross walls, we know they're called as septa. ये जो septa है, the septas can be of two types. We know in fungi we can have two different kinds of septa. One is a primary septa and one is a secondary septa. A secondary septa, or sometimes we call it adventitious septa. We call it adventitious, or we call it secondary septa, or primary septa. ठीक है, आपने याद रखना है कि जो primary septa है, primary septa is always formed in association with a nuclear division. भाई मैंने कहा ये एक सेल है हमारी, ठीक है, यहाँ पे एक ही न्यूक्लियाई है, अब इसका डिवीजन होगा, मैंने divides, we have two cells, so when we have two now, ये देखो दो न्यूक्लियाई हो गए, तो जैसी सेल का डिवीजन होगा, जैसी karyokinesis, as the nuclear division will take place, in between these two nuclei, a new septa will be formed. A new septa is formed. So such kind of septa, which is formed in association with nuclear division. मैं क्या कह रहा हूँ? Septa which is formed in association with nuclear division. This is called as what? This is called as the primary septa. This is called as the primary septa. Okay? And the septa which is formed independent of nuclear division. Septa formed independent of nuclear division. This is called as what? This is called as secondary septa or adventitious septa. For example, मैंने कहा भाई हमारा ये हाइफे है, ठीक है, ये हमारा हाइफे है, अब यहाँ पे एक फ्रूटिंग बॉडी बन गई भाई, यहाँ पे एक फ्रूटिंग बॉडी बन रही है, देखो फ्रूटिंग बॉडी बन रही है, तो इसका क्या होता है, शुरू में तो यहाँ पे देयर इज नो सेप्टा, शुरू में तो सेप्टा नहीं होता है यहाँ पे, दिस इज अ फ्रूटिंग बॉडी, बट वंस द फ्रूटिंग बॉडी फॉर्मेशन इज कंप्लीटेड, दिस फ्रूटिंग बॉडी वुड बी कट ऑफ, ये देखो यहाँ पे सेप्टा फॉर्मेशन हो गया, इट इज कट ऑफ. So reproductive organs normally they are cut from the mycelium by means of septa. Or sometimes what is happening, जैसे मैंने कहा ये हमारा hyphae. So the older hyphae में we have less cytoplasm. The new hyphae, और आपने याद रखना है कि hyphae will always grow at the tip. The growth of fungi will always take place at the tip. मतलब यहाँ से बीच में growth नहीं हो रही है. There is no growth over here. कि बीचों बीच, no growth in between. The growth of fungi will always take place at the tip. So we have older part also, older part with less cytoplasm. The new hyphae has more cytoplasm. So sometimes this older part is cut off from the new part. The older part of hyphae which has less cytoplasm, it is cut off by means of septa. Now this is independent of nuclear division. We know this is independent of nuclear division. So such kind of septa, we call them as what? We call them as secondary, or we call them as adventitious septa. So this should be very, very clear. And we know, I am talking about septa, we know this septa can be a complete structure. We know it can be a complete structure, which is a complete plate-like structure. It can be a complete plate, or sometimes we see a septa may be porous. A septa can also be porous. We can have a pore in between. So it can be a complete plate, it can be porous structure, and sometimes we see in certain members, look at this, in certain members, what we see, each septa, it forms a barrel-shaped structure. Septas can also form, look at this, they can form a barrel-shaped structure. देख रहे हो? You know, they can also form a barrel-shaped structure. In certain fungi, the septa has a barrel-shaped structure which is covered by a perforated plate. देखो, इसके चारों तरफ क्या present है? इसके चारों तरफ एक plate-like structure present है which is perforated. A plate-like structure which is perforated. So such kind of septa, very important, such kind of septa ठीक है, which is a barrel-shaped structure surrounded by a perforated membrane. We call it what? You all know this. The people of botany must have studied this. This is called as dolypore. This is dolypore septum. We call it dolypore septum. Look at this, dolypore septum. And we know dolypore septum is a characteristic feature of, सब में नहीं मिलता ये. This is a characteristic feature of basidiomycetes. This is a characteristic feature of basidiomycetes. जैसे Puccinia. Puccinia we know is a basidiomycetes. Ustilago we know is a basidiomycetes. Agaricus, मतलब mushroom, we know it is a basidiomycetes. All these are examples of basidiomycetes. Okay, so this you should know. Now, see, we have been talking about fungi, and when we talk about fungi, one important feature of fungi is the cell wall. We know cell wall. We know fungi have cell wall. Is this very clear? And we all know the cell wall of fungi contains chitin. We know cell wall of fungi contains chitin. Chitin is made up of N-acetyl glucosamine. जो chitin है, this is basically made up of repeating subunits of NAV, N-acetyl glucosamine. And we know it is a characteristic feature. This is a characteristic feature of fungi. Chitin is a characteristic feature of fungi. Most fungi, they contain chitin. But in recent years, what we have discovered that members of oomycetes, they do not contain chitin. So in recent years, what we have found, most of the members of oomycota or oomycetes, they lack chitin. Most members, but certain exceptions are there. Certain exceptions are there. For example, there's an exception called as we have apodeclia. There's a member called as apodeclia. This is apodeclia. Now this is a member of oomycetes, but it contains chitin. This member we know contains chitin. So this is an exception here. This is an exception. Okay? Otherwise, most members of oomycota, they do not contain chitin. So coming back to the cell wall, we know members of oomycetes, इनकी जो cell wall है, their cell wall mainly contains cellulose. Plus they also have beta glucans. So cellulose plus beta glucans. If you look at the ascomycetes, if you look at the members of ascomycetes member have chitin. Ascomycetes में हमारा yeast आता है, ठीक है? So we know members of ascomycetes, they have chitin, plus they also have beta glucans. ये कई बारी matching में आ जाता है. Examiner can give this to you in matching, क्योंकि cell walls are very important. So look at the cell walls of oomycetes, ascomycetes with chitin and beta glucans. Look at the members of basidiomycetes. Members of basidiomycetes, we know they have chitin plus mannan. They have chitin plus mannan. Look at the members of deuteromycetes. Look at the member of deuteromycetes. Deuteromycetes again have chitin plus beta glucans. They have chitin plus beta glucans. So look at different members of fungi. Their walls contain different components. And by the way, members of deuteromycetes, deuteromycetes, this entire class, we call them as fungi imperfecti. So what is fungi imperfecti? Fungi imperfecti. What is called as fungi imperfecti? Members of deuteromycetes. This is जैसे fusarium, polytotrichum, penicillium. They were earlier all under deuteromycetes. Penicillium, they're all members of deuteromycetes. तो आपने याद रखना है कि all those fungi whose sexual stages have not been discovered. मैं क्या कह रहा हूँ? Those fungi whose in whom sexual reproduction have not been discovered. They are all kept under fungi imperfecti, that is deuteromycetes, जहाँ पे sexual reproduction have not been discovered. ठीक है? So this should be very, very clear. So we have seen about the cell walls. Now, when we talk about fungi, let us talk about the nuclear division or cell division. Cell division in fungi. आपने याद रखना है. Now this is very peculiar. Most members of fungi, most of the members in fungi, they have closed mitosis. They show what is closed mitosis. So fungal members basically divide by means of mitosis, we know. ठीक है, but it is a closed mitosis. Now, अगर हम mitosis की बात करें, देखो, we know mitosis can be of two types. अभी आपने सुना closed, and the second is open mitosis. Open mitosis. Closed mitosis में the nuclear membrane does not break down. What is a closed mitosis? ये जो nuclear membrane है, the nuclear membrane, it does not break down. मतलब it remains intact. It remains intact. Whereas open mitosis में the nuclear membrane disintegrates. It breaks down. The nuclear membrane it basically disintegrates. Look at this. So in most fungi, there are exceptions are there. Exceptions are there. For example, certain myxomycetes. हमने अभी देखा था ना myxomycota, slime molds. So we have exceptions. We know in certain myxomycota members, we have open mitosis also. So in certain members of fungi, we have open mitosis also. But generalization, generally, they show what is closed mitosis. That means the nuclear membrane does not break down. Okay, so this should be very, very clear. Okay, so that means हमने क्या देखा है कि in many members of fungi, the spindle fibers are formed inside the nucleus. मतलब जो spindle fiber का formation होता है, that is formed inside the nucleus only. Nucleus के बाहर नहीं, अंदर ही बन जाते हैं. So we call it what is closed mitosis. ठीक है, अब एक चीज और याद रखो. So this is one type कि closed mitosis, open mitosis. Look at another classification. See, when I'm talking about nuclear division, when I talk about nuclear division, आपने याद रखना है कि nuclear division मतलब क्या बोले? बोले तो karyokinesis. Nuclear division is called as karyokinesis. हम कहते हैं ना, माइटोसिस में मतलब सेल डिवीजन में cytokinesis, karyokinesis is followed by cytokinesis. We generally say what? We say कि karyokinesis मतलब nuclear division is followed by the division of cytoplasm. So karyokinesis is followed by cytokinesis. So I am talking about nuclear division in fungi. Now nuclear division can be of two types. Fungi में we can have two different kinds of nuclear division. One is called as centric and one is called as non-centric. So two kinds of nuclear division, centric and non-centric. आपने याद रखना है कि when I say nuclear division is centric, मतलब कि we have centrioles which are present. It involves centrioles, मतलब it is characterized by presence of centrioles. So centric nuclear division, मतलब this is characterized by the presence of centrioles around the nucleus. Around the nucleus. Is this very clear? और जो centric nuclear division है, ये भी याद रखो. This centric nuclear division, ये पहला point हो गया. The centric nuclear division is found in those fungi. It is found in those fungi which produce flagellated cells. So only those fungi, very important, which produce flagellated cells. Flagellated cells any time in the life cycle, anywhere in the life cycle, anywhere in their life cycle. Okay, so they are characterized by centric nuclear division. They show what is centric nuclear division. तो आपने याद रखना है कि सारे फंजाई जो होते हैं, ऑल दी मेंबर्स ऑफ फंजाई डू नॉट प्रोड्यूस फ्लैजिलेटेड सेल्स. जैसे मैं एस्कोमाइसीड्स की बात करूं, यीस्ट, एस्कोमाइसीड्स में आप पढ़ते हैं यीस्ट वगैरह, तो मेंबर्स ऑफ एस्कोमाइसीड्स दे हैव नो फ्लैजिलेटेड सेल्स इन देयर लाइफ साइकिल. फ्लैजिलेटेड सेल्स तो होते नहीं वहां पर. बेसिडोमाइसीड्स नो फ्लैजिलेटेड सेल्स, डिट्यूरोमाइसीड्स नो फ्लैजिलेटेड सेल्स. तो भैया मतलब 70% फंजाई में तो फ्लैजिलेटेड सेल्स होते ही नहीं है. दे ओनली हैव नॉन फ्लैजिलेटेड सेल्स. फॉर एग्जांपल मैंने कहा ना नॉन फ्लैजिलेटेड सेल्स. तो फिर फ्लैजिलेटेड सेल्स किसमें? फ्लैजिलेटेड सेल्स फॉर एग्जांपल वी हैव oocytes. Members of oocytes or members of oomycota, they show flagellated cells. They have flagellated cells in their life cycle. For example, we have zoospores. We have zoospores. We call them as zoospores. Flagellated cells. Okay. So all the fungi will not produce flagellated cells. Certain members of myxomycota. अभी हमने बात की थी slime molds. Slime molds. Myxomycota. They also have flagellated cells. Their cells also contain flagellated cells. Is this very clear? So, which members of fungi will contain flagellated cells? My friend, only those fungi whose nuclear division is centric will have flagellated cells. याद रखो. Centric मतलब they are characterized by presence of centrioles around the nucleus. So this is a very important feature. ये जो feature it is very important and it is used in classification of fungi. This characteristic is very important in fungal classification. अब दूसरी तरफ देखते हैं non-centric. The name itself is very clear कि centrioles are absent. Non-centric मतलब centrioles absent. Centrioles are absent. So that means those fungi which show non-centric nuclear division, they do not produce flagellated cells. They do not produce flagellated cells in their life cycle. Is this very clear? For example, I give examples. Ascomycetes, basidiomycetes, example, asco, basidio, etc. Okay. So centrioles are not present. But instead of centrioles, we have something else. Centrioles are absent, but instead of centrioles, we have what? They have SPB. Centrioles are absent, but they have special structures. Their cells have special structures which have taken care of absence of centrioles, which take care of centrioles. We call them as SPB. We call them as double AO. ऐसे कुछ structure होता है. Just remember SPB. I just remember what is SPB. So one structure is called as what is SPB. This is spindle pole body. So they have what is spindle pole body. एक और structure होता है. Sorry, yeah. Now, so one is SPB. Their cells also contain a structure called as now. This is what? This is nucleus associated organelle. So what is now? This is nuclear or nucleus associated organelle. What is SPB? The spindle pole body. ठीक है. और दोनों ही जो structures होते हैं, they are found in the nuclear membrane itself. Both these structures are found integrated in the nuclear membrane. And they help in the formation of spindle fibers. They help in formation of spindle fibers. Okay, but centrioles are absent. So they have SPBs, they have now, which will take care of the centrioles. Okay, so this classification should be very, very important. Centric, non-centric. Centric, flagellated cells, non-centric, no flagellated cells in the life cycle. ठीक है. अब देखो, मैंने आपसे कहा था कि थैलस इज मेनली मेड अप ऑफ माइसीलियम. जो फंगल थैलस होता है Fungal thallus is mainly made up of mycelium. ठीक है, और मैंने आपसे ये भी बोला था, I also told you कि under certain conditions, we know this mycelium can be organized. This mycelium, it gets organized to form certain tissue-like structures, to form certain organized structures, certain organized tissue-like structures. देखो, and मजे की बात देखो, something which is surprising, sometimes we see कि जो tissue-like structures हैं, they also show division of labor. They also show certain division of labor. चल तू ये काम कर ले, मैं ये काम कर लेता हूँ. So they are also found to show certain division of labor also. So under certain conditions, the mycelia, the hyphae, they can get organized to form tissue-like structure. So we are looking at these organized structures now. We are looking at these organized structures which are formed by the mycelium. So mycelial, we are looking at the mycelial mycelial structures. Let us look at the mycelial structures. ठीक है, तो देखो सबसे पहले आपने याद रखना है rhizomorph. There's a structure called as rhizomorph. So certain fungi during certain stage of life cycle, they produce structures called as rhizomorph. They produce structures which are called as rhizomorph. और आपने याद रखना है कि जो rhizomorph, this is a structure to tide over unfavorable conditions. This is a structure which is formed to tide over unfavorable, unconducive, unfavorable conditions. ठीक है, अब rhizomorph में हमने देखा है, in this rhizomorph, ये जो mycelium है, this mycelium basically forms thick strands. ठीक है, अगर हम इस rhizomorph को देखेंगे, तो rhizomorph में you will see thick strand-like structures. The mycelium forms thick strand-like structures, और जो individual hyphae हैं, the individual hyphae lose their individuality. तो जो unit hyphae हैं, the unit hyphae lose their individuality. It loses their individuality. So they tend to lose their individuality. You cannot identify individual hyphae over here. Okay? And they form complex tissue-like structures. So in rhizomorph, what we see is a complex tissue-like structure, which shows division of labor. This also shows division of labor. This basically shows division of labor. ठीक है, अगर आप स्ट्रक्चर को देखोगे माइक्रोस्कोप में, if you look at the structure under microscope, it is basically very similar to a root tip. The structure is basically very similar to a root. जो root होती है, this structure is very similar to a root tip. It has a growing tip also. यहाँ पे एक growing apex है. This is a growing tip. It shows a growing tip. Plus, it also has hard cortex. ये cortex है सारा. This is a very hard cortex. It also has a hard cortex. ठीक है, और ये जो structures होते हैं rhizomorph, I have just told you that these structures are formed to resist, to tide over. Tide over मतलब resist. To tide over, to resist unfavorable conditions. Unfavorable conditions. And when the conditions become normal, when the conditions become favorable, they will start again germinating. The growth is resumed. They start germinating. The growth is resumed. ठीक है, और जो rhizomorphs है, normally these rhizomorphs are more commonly found in basidiomycetes. So they are commonly found in basidiomycetes. देख रहे हो, मतलब mycelium can also form certain tissue-like structures. So one of the structure is a rhizomorph. और देखते हैं, अब एक आपने स्ट्रक्चर याद रखना है, which is called as प्लेक्टेनकाइमा। प्लेक्टेनकाइमा, तो भाई ये प्लेक्टेनकाइमा क्या होता है? अब याद रखो कि ड्यूरिंग सर्टेन स्टेज इन लाइफ साइकिल द माइसीलियम इज ऑर्गेनाइज्ड इनटू लूजली और कॉम्प्लेक्सली वोवन स्ट्रक्चर। तो व्हाट इज अ प्लेक्टेनकाइमा? प्लेक्टेनकाइमा में जो माइसीलियम है, this mycelium is organized to form loosely or compactly woven structure. So it is a loosely or compactly woven structure during certain stage of life cycle, and we call it what is प्लेक्टेनकाइमा। we call it what is प्लेक्टेनकाइमा। ठीक है, सो वेरी क्लियर। अब आपने याद रखना है ये जो प्लेक्टेनकाइमा है, दिस प्लेक्टेनकाइमा कैन आल्सो बी ऑफ टू टाइप्स। दिस कैन बी ऑफ टू टाइप्स। मैंने कहा ना लूजली, compactly. So we can again classify this into two types. One is called as prosenchyma. One is called as prosenchyma, and the second is called as pseudoparenchyma. Pseudoparenchyma. So two types of plectenchyma we have. One is prosenchyma, one is pseudoparenchyma. Prosenchyma मतलब loosely woven structure. Or in this loosely woven structure, you can identify the individual hyphae. So individual hyphae have not lost their identity. याद रखो ये जो hyphae हैं, hyphae can be identified and they lie parallel to each other. They basically lie parallel to each other. So they have not lost their identity. So this loosely woven structure, which is prosenchyma, this prosenchyma is a type of plectenchyma. Now, दूसरा क्या है? Pseudoparenchyma. Pseudoparenchyma में हमने याद रखना है कि the hyphae are so compactly packed. ये जो hyphae है, ये जो mycelia है, this mycelia is so compactly packed. It is highly compactly packed. कि individual hyphae cannot be identified. यहाँ पे जो individual hyphae हैं, they have lost their individuality. You cannot identify them. They cannot be distinguished. ठीक है? And when you look at a pseudoparenchyma में you can see कि the structure is made up of, tightly packed isodiametric cells. Tightly packed isodiametric cells. Look at this. You cannot see elongated hyphal structures. They have lost their individuality, and you can see closely packed, tightly packed isodiametric structures or isodiametric cells. And these isodiametric cells resemble parenchyma cells. They start resembling parenchyma cells. और तभी इसको कहते हैं pseudoparenchyma. Is this very clear? ठीक है? तो we have plectenchyma, prosenchyma, and pseudoparenchyma. And we know it is nothing else. It is a loosely or compactly woven structure. Now, ये जो plectenchyma है, this plectenchyma can be used. It is basically used to form different somatic and reproductive structures. You know, plectenchyma, ऐसे थोड़ी बता रहा हूँ मैं आपको plectenchyma. Plectenchyma can be used to form either reproductive structures or somatic structures. Somatic or reproductive structures. अब कौन से ऐसे structures होते हैं, ये important है. Which structures are formed of plectenchyma? So we have two such important structures which are formed out of this plectenchyma. One is called as stroma. One of the structures is called as what? One of the structures is called as stroma, or sometimes we call it stromata. Stroma or stromata. The second, which is more important, is called as sclerotium. Sclerotium. So two structures can be formed. One is stromata, one is sclerotium. Is this very clear? ठीक है, ये जो स्क्लेरोशियम है आपने याद रखना है, this is a hard resting body. For sclerotium, it is a very hard resting body. This is nothing. This is rhizome था ना. Similarly, this is also a resting body to resist unfavorable conditions. ठीक है, और जो स्ट्रोमेटा है, स्ट्रोमेटा is a cushion shaped structure. This is basically a compact cushion shaped structure on which fructifications are normally born. So cushion shaped structure on which the fruiting bodies are normally found. This is stromata. But sclerotium is what? It is a very complex structure to tide over unfavorable conditions. So it is nothing else, it is a resting body. So we are looking at general aspects of fungi. ठीक है, क्योंकि एक शब्द दे रखा है छोटा सा कहीं छुपा के उन्होंने फंजाई, तो हमने सोचा चलो कर लेते हैं यार, लेट्स नॉट लीव इट, क्या पता एक क्वेश्चन बच जाए, आपका एक क्वेश्चन कहीं से आ जाए, हो सकता है, rather than leaving it, let us discuss. ठीक है, so अब पूरा सब्जेक्ट नहीं डिस्कस कर सकते क्वेश्चन के लिए, it will take multiple lectures, शायद we can have 15 to 20 lectures, मतलब we can discuss for 20 to 30 hours. फंजाई तो बहुत बड़ा है, बल्कि बॉटनी वालों के लिए फंजाई इज द मोस्ट कॉम्प्लिकेटेड सब्जेक्ट जो बच्चों को समझ में नहीं आता. ब्रायोफाइट्स इज इजी, एल्गे इज इजी, टेरिडोफाइट्स इज इजी, फंजाई इज मोर डिफिकल्ट. मुझे सबसे डिफिकल्ट जो है वो फंजाई है. इतनी वैरायटी है फंजाई में, normally we don't study in colleges. Now the question is, see, we have talked about different kinds of tissue structures, we have talked about nuclear divisions, the cell divisions, all that we have talked about. Now when I talk about the optimal conditions of growth, अगर फंजाई के हम ऑप्टिमल कंडीशंस की बात करें ग्रोथ की, ग्रोथ के लिए व्हाट आर द ऑप्टिमल कंडीशन? जनरली फॉर मोस्ट फंजाई व्हाट आर द ऑप्टिमल कंडीशंस ऑफ ग्रोथ? आपने याद रखना है द ऑप्टिमल पहले जो टेम्परेचर होता है, टेम्परेचर इज अराउंड 20 to 30 डिग्री सेंटीग्रेड. दिस इज सेड टू बी द ऑप्टिमल टेम्परेचर फॉर ग्रोथ, 20 to 30 डिग्री सेंटीग्रेड, नॉट वेरी हाई टेम्परेचर. ठीक है, दूसरी चीज आपने याद रखनी है, ये जो फंजाई है, फंजाई नॉर्मली प्रेफर एसिडिक मीडियम फॉर ग्रोथ. They prefer acidic medium. ये याद रखो आप, for their growth. Normally, the pH should be something around 6, slightly acidic. pH, suitable pH is something around 6. ठीक है, तीसरी चीज आपने याद रखनी है, the third thing you have to remember, कि light is not essential for fungal growth. Light is not essential for fungal multiplication or fungal growth. Not essential. क्योंकि एक क्लोरोफिलस है, आपको पता है, so light is not essential. But मैंने कहा ना for multiplication or growth, but light is important. For fungi, this light is important for sporulation. मतलब spore formation, light is required. ठीक है, so this you have to keep in mind. Okay. अब एक चीज और याद रखो कि in fungi which have septa, तो fungi whose hyphae have septa, जिनमें septa होता है, जैसे मैंने कहा ना oomaisids में the hyphae sinocytic, there is no septa, it is aseptate, multinucleate. मैं उनकी बात नहीं कर रहा हूं, I am not talking about oomycota, I am talking about fungi whose hyphae are septate. What we have seen, at their tip, in these fungi normally at their tip, they have a refractive body. यहाँ पे एक refractive body है. They are found to have a refractive body, and this refractive body is called as Spitzenkorper. क्या नाम है इसका? This is Spitzenkorper. This is Spitzenkorper. This is normally found near the fungal apex. What is the use? No idea. इसका काम क्या है हमें नहीं पता. What is the use of this? ठीक है. Second thing that you have to remember, I just told you कि fungal growth, when I'm talking about the fungal growth, तो मैंने I just told you fungi will always grow at the tip. The growth is always at the tip. The growth is always at the tip. ठीक है, मतलब अगर हम टिप की बात करें तो बोले तो मतलब क्या हुआ, मतलब ये हुआ, just hold on. What do you mean is when I look at this cell, भाई हमारा न्यूक्लियस है, ये तो हमारा न्यूक्लियस है, लेकिन यहाँ पे एंडोप्लाज्मिक रेटिकुलम भी है क्योंकि यूकैरियोटिक है. We know fungi as eukaryotic. Eukaryotic है तो that means what? They will have ER, they will have भाई Golgi bodies भी होंगे यहाँ तो. They'll also have Golgi bodies. तो भाई ER है तो यहाँ पे क्या होगा? प्रोटीन सिंथेसिस होगा, सब कुछ होगा. Vesicles बनेंगे यहाँ पे. Vesicles will bud off from here. Vesicles will fuse with this. यहाँ से vesicles बनेंगे. And these vesicles which bud off, these vesicles which bud off from ER, from Golgi bodies, they will carry the material. They carry the material for the fungal growth. ये आके यहाँ से tip से fuse कर देंगे. They'll fuse with this tip. बात समझ में आ गई? And they will give the material for the growth of the fungal cell. So they carry the material for the growth of the fungal cell. So the growth is by means of what? The growth is by means of, vesicular. So what do we call this as? This is called as vesicular hypothesis of growth. क्या कहते हैं इसको? This is vesicular hypothesis of growth. ये सब आपको याद रखने की जरूरत नहीं. एक ही तो क्वेश्चन आना है यार, उसके लिए इतने hypothesis you don't need to remember, but just for your knowledge, vesicular hypothesis of growth. So this is also clear. Now, see, when we talk about the nutrition, fungal nutrition, पहली चीज तो हमें पता है भाई, खाना तो बना नहीं सकते क्योंकि they don't have chloroplast, non photosynthetic. मैंने बताया था they are achlorophyllous. We know they are basically achlorophyllous. So that means because they cannot synthesize food, मतलब they are heterotropic. Autotropic नहीं है, they are not autotropic. मतलब क्या? मतलब they are what? They are heterotropic by nature. They are heterotropic. So fungi are heterotropic. ठीक है, अब heterotropic भाई दो तरीके के हो सकते हैं, two types. Either they can be parasites, खाना चाहिए भाई बना नहीं सकते. So either they can be parasites. हमें पता है parasites मतलब they will grow on living bodies. They need living tissues for survival. So either they can be parasites or they can be saprobes, saprophytes. They will grow on dead organic matter. जैसे लकड़ी हो गई, लकड़ी में we know we have bracket mushrooms going, cow dung. कई बार आप देखते हो cow dung में से mushrooms निकल जाते हैं, जो गोबर होता है. क्या है वो? तो dead material है ना, dead organic matter. So fungi can be either parasites or they can be saprobes. अब parasites भी बोले तो they can be of two types, facultative or they can be obligate. Parasite भी दो तरीके हो सकते. Similarly saprophytes two types, they can either be facultative or they can be obligate saprophytes. ठीक है. So when I talk about facultative parasites, याद रखो, facultative parasites are those saprophytes. Facultative parasites are those saprophytes which have the capacity to infect living plants or animals, which have capacity to infect or to grow living plants or animals. They can grow on living plants or animals. Parasites, facultative parasites are actually saprophytes which have the capacity to infect living plants or animals. और जो obligate होते हैं, obligate मतलब क्या? Obligate मतलब कि they will only live on living protoplasm. They will only live on a living cell, living protoplasm, living protoplast. So they need a living cell. Same अगर हम saprobes की बात करें, if I'm talking about facultative saprobes, we know facultative saprobes are actually parasites. So those parasites which have the capacity to grow on dead organic matter. So parasites which have the capacity to grow on dead organic matter. In the absence of living host, they can grow on the dead organic matter also. So we call them as facultative saprophytes. बाकी obligate मतलब तो पता है आपको, obligate मतलब they'll only grow on the dead organic matter. They only grow on the dead organic matter. ठीक है? So this is broadly about the fungal nutrition. ठीक है? Either they're parasites or they're saprophytes. Now, let us talk about the reproduction. Fungal reproduction. अब ये इंपॉर्टेंट हो जाता है. रिप्रोडक्शन तो इंपॉर्टेंट होती है अपने आप में, है ना. तो जब हम रिप्रोडक्शन की बात करें, हमें पता है कि फंगल रिप्रोडक्शन कैन बी आइदर सेक्सुअल और दिस कैन बी एसेक्सुअल, बोथ. इट कैन बी सेक्सुअल एज वेल एज एसेक्सुअल. ठीक है. अब बिफोर वी टॉक अबाउट रिप्रोडक्शन, we have to know something. There are two terms which are important. There are two terms, दो ऐसे शब्द हैं that we need to know about them. One is called as holocarpic and one is called as eucarpic. So if I use the term holocarpic fungi, eucarpic fungi, इसका क्या मतलब हुआ? What does it mean? Holocarpic, eucarpic. Holocarpic मतलब, when I say holocarpic fungi, that means that the entire thallus is converted into reproductive structure. So in a holocarpic fungi, the entire thallus, this is converted into a reproductive structure. So we call such fungi as holocarpic. मतलब कहने का ये, I can also say like this, कि somatic and reproductive phase, they do not coexist. क्योंकि entire thallus is converted to reproductive structure. तो मतलब जो somatic phase है, somatic phase and reproductive phase, they do not occur together. मतलब they do not coexist. They do not coexist. Holocarpic देखो. और जब हम eucarpic की बात करते हैं, when we talk about the eucarpic fungi, in the eucarpic fungi, we know only a part of thallus is converted to the reproductive structure. Only a part of thallus. मुझे लिखने की जरूरत नहीं है सब कुछ. You are smart enough to decipher what I say. Only a part of thallus gets converted to reproductive structure. मतलब कहने का ये, what I try to mean is that reproductive and somatic structures can coexist. तो ये जो reproductive structure है और जो हमारा somatic structure है, they can coexist. So this is what is eucarpic. Now, so we know now holocarpic fungi, eucarpic fungi. The question is, let us talk about first asexual reproduction, modes of asexual reproduction in fungi. ठीक है, तो भाई हम कह सकते हैं फिजन, ये भी मोड है हमारा. हम कह सकते हैं बडिंग, ये भी हमारा मोड है. हम कह सकते हैं asexual reproduction by means of arthrospores. Arthrospores. D. Asexual reproduction by means of chlamydospores. E. Asexual reproduction by means of spores. Spores. So these are the basic methods of asexual reproduction. Let us talk about them one by one. Basics. Fission. Fission बोले तो simple cell division. This is simply a very simple somatic cell division. Simple cell division. Cell है, nucleus, cell grows, elongates, nuclear also elongates, divides into two. हमारी cell है, nucleus again divides, elongates, divides into two, and now karyokinesis has taken place. This is followed by cytokinesis, and we get two cells. We get two cells. So fission matlab is a simple cell division, nothing else. This is what is fission. Budding, budding बोले तो भाई हमारा cell है, हमारे cell में there's a bud which is formed. There's a bud formation starts. A bud formation starts. Nucleus था, nucleus divides. Nucleus है, nucleus divides into two. One of this nuclei will percolate into the bud. One of this nuclei goes into the bud. देख लो, bud grows in size. अभी हमारा जो bud it grows in size. Nucleus अंदर आई गया है यहाँ पे देखो. And there's a continuity of cytoplasm. We can see there's a continuity of cytoplasm between the bud and the mother cell. ये हमारी mother cell, ये bud है हमारा. ये हमारा nucleus है. And after some time, what will happen? ये जो continuity है of cytoplasm, this is closed. This is closed. और ये जो bud है, bud will be removed from the mother body. हमारी mother body और ये bud है, this will be separated, and we get two individual cells. Sometimes the bud may also remain intact. ऐसा नहीं कि we have conditions where the buds which are formed do not get separated from the mother cell. But in many cases, the buds will get separated and they form two individual cells. So commonly, this is found in the case of yeast. Yeast. We see what is budding. ये सब आप 11th 12th क्लास में पढ़ते हो. What is fission? What is budding? Fission भी कई तरीके की होती है. Okay. Now the third is more important. Arthrospores. What is arthrospores? अब देखो. मैंने से arthrospores, मतलब arthrospores में जैसे मैंने कहा ये पंजा है हमारा, fungal hyphae. Look at this fungal hyphae. I said this is a fungal hyphae. ये हमारा fungal hyphae है. अब क्या होता है? In this arthrospores, formation of arthrospores, what is happening? This fungal hyphae will break into component cells. This fungal hyphae breaks into component cells. एक cell यहाँ बन गई, एक cell अलग हो गई यहाँ से, ये cell अलग हो गई. The cells get separated. The individual cells get separated. And they start behaving like what? Behaving like spores. They start behaving like spores. Individual cells get separated and they behave spores. Again, they'll start multiplying. So this is what is arthrospores. And what is a chlamydospore? What do you mean by chlamydospores? Chlamydospore में आपने याद रखना है, how it differs from the arthrospores is that before separation, the fungal hyphae, the cells, and they become thick walled. So when I say chlamydospores में the cells, before they start separating, they become thick walled. The walls will become thick. And then they get separated. So the prerequisite is that the cells, इनकी जो walls हैं, the walls become thick. They're thick walls. And then they get separated. And then they separate. They separate. So cells will separate have thick walls. Thick walls, cells will get separated. Okay, and they form what is chlamydospores. So thick wall spores, thick walled spores, we call them as chlamydospores. This is what is chlamydospores. And we know the chlamydospores are used to tide over unfavorable conditions. They are used to resist, to tide over unfavorable conditions. So which spores are used to tide over unfavorable conditions? Arthrospores or chlamydospores?
Chlamydospore.
चलो पूरे लेक्चर में एक आवाज तो सुनने को मिली. नहीं तो इतनी देर से नहीं बोले जा रहा हूं. Now let us come down to the last one, which I say spores. देखो I'll take a new page. So when I say spore formation, अभी तक देखो कोई actual spore formation नहीं हुआ है. अभी तक तो सिर्फ हमने cells get separated. Some become arthrospores, some become chlamydospores. But formation of specific spores. Okay. Now, two types. The actual spores are taken into two types. पहला, the spores which are born inside sporangia. Spores which are born inside sporangia. Sporangia क्या होते हैं? Sporangia are fruiting bodies. They are nothing else. They are fruiting bodies. जैसे मैंने कहा, ये हमारा एक sporangia है, ये हमारा sporangia. ठीक है? यहाँ पे spores है. Not a spores over here. This is a fruiting body. This is a fruiting body with multiple spores. So when spores are born inside this sporangia, this is one method, and we call them as what? We call such spores as what? We call such spores as sporangiospores. क्या कहते हैं? Spores born inside sporangia, we will call them as sporangiospores. ये देखो sporangiospores. ठीक है? तो ये क्या है? Sporangiospores. ये sporangia है हमारा. और ये क्या हमारे sporangiospores है हमारे. Spores, sporangiospores. There's one thing I have to tell you, something I have to bring to your kind notice, my lord. The sporangiospores can be of two types. They can also be of two types. One which is a motile and one which is non-motile. One which is motile, one which is non-motile. तो भाई जो sporangiospores हैं, released flagellas appear, they become motile. And motile sporangiospores, the motile sporangiospores हैं, we call them as what? We call them as zoospores. We call them as zoospores. So zoospores are nothing else. They're motile sporangiospores. We call them as zoospores. They are flagellated. और मैंने आपको पहले ही कहा था कि all the fungi do not produce flagellated cells in the life cycle. Only those fungi which have centric nuclear division. याद है कि भूल गए? बताओ मुझे.
Yes.
याद है ना? Only those fungi which show centric nuclear division will produce flagellated motile spores. For example, mycota. ठीक है? दूसरी तरफ, when these sporangiospores are non-motile, when they are non-motile, we call them as what? We call them as aplanospores. So non-motile sporangiospores, we call them as aplanospores. Is this clear? They are non-flagellated, non-motile. So this is the first class. Spores. We're talking about the sporangiospores. अब second type देखो. मतलब यहाँ पे तुमने पहले देखा ना कि born inside sporangia. अब दूसरा type देखो. The second type of spore, the second classification, when the spores are born on the tip of a hyphae. ये देखो, ये hyphal tip है. Tip. So hyphal tip will start cutting spores. When the hyphal tip starts cutting spores, मतलब produced at the tip of hyphae, we call them as what? We call such spores as are called as conidia. We call them as conidia. जैसे मैंने कहा example, albugo, peronospora, albugo, mucor, क्या है सब? ठीक है? They all cut off spores at the tip of the. So we call them as conidia, or sometimes we call them as conidiospores. Conidia बोल लो चाहे conidiospores बोल लो. One and the same thing. ठीक है? So this should be very very clear. तो कई बार हमें याद रखना है कि aplanospore मतलब नॉन मोटाइल. तो चाहे वो sporangiospores हो चाहे conidia हो, because they are all non-motile. So any non-motile spores in general we call them as aplanospores. So sometimes conidia will also come under air panospores only. So this is about. So if you look at the zoospores, मैंने कहा zoospores are flagellated. ठीक है, तो मैंने से flagellated, यहाँ पे flagella है, यहाँ पे भी है flagella. So if they have flagella, मतलब they will have flagellar apparatus also. They will have flagellar apparatus. तो कई बार फंजाई में क्या होता है, फ्लैजुलर apparatus से क्वेश्चंस आ जाते हैं. They will start asking questions from flagellar apparatus. तो very simple. मैंने कहा भाई ये फ्लैजुला है हमारा, ये मेरा फ्लैजुला है. Nothing else. Flagella we know is appendage. ठीक है, grows out from the cell. और फ्लैजुला जो होता है, it is nothing else. It is extension of plasma membrane. देख लो, plasma membrane का extension हो गया. बस इसके अंदर में क्या है? Inside this, what is the core? The core is here. Look at the core. This is the core of this flagella. The flagella has a core, and this core is called as exoneme. So core of flagella or cilia. देखो flagella and cilia broadly, they have the same structure. They have a core which is called as exoneme. और exoneme किसकी बनी है? This exoneme is made out of microtubules. Exoneme is made out of अगर मैं इसका TS काटूं, if I cut the section of this transverse section, I'll see this kind of structure. ठीक है, और यहाँ पे सेंटर में क्या है? सेंटर में we have microtubules. देख रहे हो? ये देखो microtubules, microtubules, microtubules. ये देखो microtubules. So how many? 9 periphery and 2 in the center. 2 in the center. So 9 periphery वाले आप देख रहे हो? They are what? They are doublets. So 9 peripheral doublets. Center वाले 2 singlets. In the center 2 we have singlets, and that is why we call this arrangement as what? ये जो exoneme है, this is arrangement of 9 plus 2. So 9 plus 2 arrangement of microtubules. It has a 9 plus 2 arrangement of microtubules. My friend and countrymen, 9 doublets which are the periphery and 2 complete microtubules in the center. ठीक है, ये एक exoneme हो गया. अब इसके exoneme कहां से निकल रहा है भाई? ये exoneme arises out of something. At the base of exoneme, ये देखो, at the base of exoneme we have what? We have a kinetosome. क्या कहते हैं इस स्ट्रक्चर को? Kinetosome. Or sometimes it is also called as a basal body. Or sometimes this is also called as a blepharoplast. सभी नाम बता दिए मैंने आपको, अलग-अलग किताबों में अलग-अलग तरीके से दे रखा है. So we have kinetosome, basal body, blepharoplast, which is present at the base of the exoneme. And it is from the basal body that the exoneme arises. Exoneme basically arises. Is this clear? अब देखो, ये हमारा न्यूक्लियस है, यहाँ पे ये हमारी सेल है पूरी. This is the cell that we have, and there's a nucleus over here. This is the nucleus. अभी जो kinetosome है, this kinetosome is also attached to the nucleus. Kinetosome is attached to the nucleus by a rootlet. ये देखो, kinetosome is attached to the nucleus by means of a rootlet. Rootlet. Sometimes this rootlet is also called as a rhizoplast. Sometimes we call it a rhizoplast. Blepharoplast nahi, rhizoplast. This is rhizoplast. So it is attached to the nucleus by means of a rhizoplast. Is this clear? This is what is the flagellar apparatus which is found in the fungi. This is they are the components of flagellar apparatus. So see, we have talked so much. We can't expect me to discuss life cycles of so many fungi just for a single word. Okay. Now the question is, you've seen so many things. We have talked about the asexual reproduction modes. Now, before we go on to the sexual reproduction, before we talk about the sexual reproduction, Let us talk about the fungal life cycle. The life cycle of fungi. ठीक है, हमें पता है भाई, देखो अगर हम लाइफ साइकिल की बात करें, हमें पता है इन लाइफ साइकिल वी हैव a haploid thallus. Sometimes we can have a diploid thallus. We can have a diploid thallus. और ये आपस में ऑल्टरनेट करते हैं. भाई मैंने कहा हैप्लॉइड थैलस है, चलो एक हम ऐसी बात कर लेते हैं, ब्रायोफाइट. मैंने कहा हैप्लॉइड थैलस है, मतलब एन, दिस इज एन हैप्लॉइड थैलस. यहाँ पे क्या बनेगा, यहाँ पे हमारे गैमीट्स बनेंगे. दिस विल गिव राइज टू गैमीट्स. दिस विल फॉर्म गैमीट्स. गैमीट्स वुड बी फॉर्म्ड ओवर हियर. गैमीट्स मतलब गैमेटेंजिया. स्ट्रक्चर्स कॉल्ड एज गैमेटेंजिया. ये हमारे स्ट्रक्चर्स है गैमेटेंजिया. यहाँ पे गैमीट्स विल बी फॉर्म. मेल गैमीट विल हैव अ फीमेल गैमीट. ये मैंने कहा चलो हमारा फीमेल गैमीट है, ये हमारा फीमेल गैमीट है. हमारे मेल गैमीट्स हैं मेल, ये हमारे फीमेल गैमीट है. मतलब दे आर ऑल एन. मेल गैमीट भी एन होगा, ये भी एन होगा, थैलस भी एन. सो दिस इज अ हैप्लॉइड जनरेशन. वी नो दिस इज अ हैप्लॉइड जनरेशन. अब क्या हो जाएगा, अब क्या होगा भाई, मेल एंड गैमीट्स आर रिलीज्ड. इनका क्या हो जाएगा आपस में, दे विल गो एंड दे विल फर्टिलाइज. n plus n will fertilize. मतलब male and female will fertilize. You will get what? You will get zygote. You will get zygote, which is the first 2n structure. First diploid structure is a zygote. Zygote starts multiplying. Multiply. We get embryo. Embryo still multiplies. We get again a thallus. ये देखो, again we get a second thallus in the life cycle. तो ये वाला हमारा पहला thallus था, which is n. And now after fertilization, zygote multiplies, we get a second thallus, which is 2n. This is diploid. 2n. All these cells are diploid. अब क्या हो जाएगा, यहाँ पे क्या होगा, in this diploid thallus what will happen, meiosis will take place. Meiosis will take place, and out of this meiosis some spores would be formed. The spores are formed, and all these spores, because they are meiotic products, they are all n. Spores are n. What will happen, these n spores will go and they'll germinate into n spores, and they'll start germinating. They start germinating and they'll give rise to what? They will give rise to haploid generation. They give rise to haploid generation. Very simple. So in this life cycle, you can see we have both the members which are dominant. Haploid is also representing, and diploid individual is also representative of the life cycle. So such kind of life cycle where haploid thallus is alternating with diploid thallus. अब मैंने बहुत सिंपल टर्म्स में आपको बताया, देखो ये, क्योंकि मेरी कॉम्प्लेक्स बताने की आदत ही नहीं. I hit at the basics. So such kind of life cycle you can see here where haploid and diploid, they're both representing the life cycle and they alternate with each other. They're alternating with each other. Such kind of life cycle is called as what? This is called as diplobiontic. क्या कहते हैं भाई इसको? इसको कहते हैं diplobiontic. Diplobiontic life. Now in a life cycle, if we only have one member which is dominant, मैंने कहा diploid हमारा है नहीं, और मैंने कहा haploid नहीं है, चलो haploid नहीं है. जैसे प्लांट्स में क्या होता है, प्लांट्स में जो हैप्लॉइड मेंबर है दैट इज ओनली रिप्रेजेंटेड बाय अ पॉलन ग्रेन. पूरा प्लांट तो डिप्लॉइड होता है ना, एंथर्स में पॉलन्स बनते हैं, पॉलन्स के टाइम पे मेयोसिस होती है, बस पॉलन्स बन गए, पॉलन्स फर्टिलाइज हो जाते हैं एग के साथ जाके. तो पूरी लाइफ साइकिल में प्लांट की एन कहाँ पे है, हैप्लॉइड कहाँ है? द हैप्लॉइड इज ओनली रिप्रेजेंटेड बाय एग इन स्पोर, दैट इज पॉलन ग्रेन. प्रॉपर थैलस तो कहीं नहीं है. आप मुझे बताओ प्लांट में कहीं हैप्लॉइड थैलस है? टेल मी यस और नो.
No, sir.
पूरे प्लांट में हैप्लॉइड थैलस तो है ही नहीं कहीं पे. The haploid face is only represented by one cell, which is pollen grain, one cell which is egg.
Yes, sir.
So what kind of life cycle is plant having? It is dominantly what is diploid. So if we have such a life cycle, in which either we have a haploid representing the life cycle, or we have a diploid face which is representing the life cycle. Only one of them, either it is haploid or diploid. Such kind of life cycle, which is only represented by a haploid or diploid, we call such life cycle as haplontic. This is haplontic life cycle, or sometimes we call it haplobiontic. कई बार haplontic कह देते हैं, कई बार haplobiontic कह देते हैं, कई बार इसको भी हम डिप्लोबायोटिक और डिप्लॉन्टिक ऐसे भी कहते थे, डिप्लॉन्टिक, डिप्लोबायोटिक. So I think so now these two terms are very clear. When I say diplobiontic, so what comes to your mind? Both the members are representative of the life cycle. When I say haplontic, matlab either a diploid is dominant or a haploid is dominant. Both will not coexist. Okay? So this should be very, very clear. और अब मैं आपको ये भी बता दूं कि diploid life cycle कहाँ होती है? Fungi में जो most fungi, they have a thallus, which is N. याद रखो. In most fungi, we have thallus, which is mainly N. It is only in Mycota. याद रखो, Mycota is an exception. Mycota, mycetes में the dominant phase of the life cycle is actually 2N. It is diploid. Mycota में life cycle is basically diploid. The dominant phase is diploid. Okay? So now we know about two different kinds of life cycles. Okay? अब देखो, let us come down to the methods of sexual reproduction. What are the methods of sexual reproduction in fungi? Okay? So primarily we have 5 methods. There are 5 different methods of sexual reproduction. One is called as planogametic copulation. Okay. Second, we call it gamete-angel contact. Okay. Third, we call it gamete-angel copulation. Sometimes gamete-angel contact is also called as gamete-angel. So मैं ऐसे भी कह देते हैं gamete-angel. Fourth one is called as spermatization. This is spermatization. And the fifth method, this is called as somatogamy. मेरा काम है बताना, याद करना आपका काम. How you remember this? That is your headache. My job is to give you clear information. So we have 5 main methods of sexual reproduction in fungi. When I say planogametic copulation, what are planogametes? Planogametic, matlab it involves what? It involves planogametes. मैंने आपको क्या कहा था कि spores which have no flagella उनको क्या कहते हैं? Aplanospores, याद है? Spores without flagella we call them as aplanospores, asexual reproduction में.
Yes, sir.
And spores which have flagella we call them as zoospores. पता है था?
Yes, sir.
Spores which have flagella we call them as zoospores. So zoospores are also called as planospores. याद रखो. Sometimes zoospores are also called as planospores. तो planogametes क्या हो गए? Planogametes मतलब the name itself is very clear कि जो zoospores हैं acting as gametes. What are planogametes? Zoospores which act as gametes. Zoospores acting as gametes. We call them as planogametes. We call them as planogametes. अब देखो मतलब planogametic copulation. Very clear. Planogametic copulation में 2 such planogametes will come together. 2 such planogametes, they come together. Okay? But they are structurally same. याद रखो. Structurally you cannot demarcate them. So structurally they are same. They come together. conjugation will take place. ये देखो, they will attach, they will conjugate, and finally zygote formation will take place. Zygote in nucleus होता है, finally zygote formation will take place, and flagella are shed, and finally we get a zygote. So structurally these two gametes are similar, but physiologically they differ physiologically. They show physiological differentiation. So finally we get zygote, which is a 2n structure. So this is what is very simple planogametic copulation. Well, the planogametes are involved, nothing, and we get a zygote after fertilization. Okay. The next one is what? Next one is gametangial contact. Okay. So again very simple. My gametangial contact में क्या होगा? Two gametes will come in contact. ये देखो, मैंने कहा भाई हमारा ठीक है, ये हमारे देखो क्या होता है, ये हमारा male gametangia. This is a male gametangia which has multiple gametes. This is a female gametangia. ठीक है, क्या हो जाएगा? ट्यूब लाइक स्ट्रक्चर्स विल ग्रो. यहाँ से देखो, ट्यूब लाइक स्ट्रक्चर विल ग्रो फ्रॉम मेल गैमेटेंजिया. अ ट्यूब लाइक स्ट्रक्चर विल ग्रो एंड मेक कांटेक्ट विद द टिप ऑफ द फीमेल गैमेटेंजिया. यहाँ जाएगा and make contact with the tip of the female gamatangia, and finally what will happen? One or more nuclei or one or more male gametes. इसमें क्या है? These are all male gametes will flow into this copulation tube. We call it copulation tube. They will grow. They'll lie in the female gamatangia, fertilize. यहाँ पे fertilization होगी, ठीक है. One or more zygotes may be formed. ठीक है, one or more zygotes may be. So this is what is sexual reproduction by gametangial contact. Sexual reproduction by gametangial contact मतलब male and female gamatangia will make contact, and nucleus of the male passes through the fertilization tube, goes and fertilizes the female gamete, or sometimes we call it egg, female gamete or the egg. A zygote will be formed. This is very clear. Sometimes we call this zygote also as oospore. Sometimes this zygote is also called as oospore. बात खत्म. So this is what is gametangial contact. What about gametangial copulation? ये देखो, gametangial copulation में क्या होगा? Very similar. Male and female gametangia will make contact with each other. ये देखो, खासकर हमने राइजोपस में देखा, जैसे ब्रेड मोल्ड होता है ना, ब्रेड में, वहां पे we see gametangial copulation. जैसे यहाँ से एक यहाँ से ट्यूब आई, यहाँ से आया. This is a positive strain, or male we can say, बल्कि we cannot say male and female over here. Male and female हमें नहीं पता होता है, ठीक है. We simply say a positive strain, one is having a negative strain. See this, they make contact, ठीक है. As they make contact, what will happen? The common wall will dissolve. क्या हो जाएगा? Fertilization will take place, ठीक है. यहाँ पे क्या हो जाएगा? ये जो gametes हैं, they will fertilize, and after fertilization, टिप पे यहाँ पे देखो जहाँ फ्यूजन हो रहा है वहीं fertilization होगा, and after fertilization what will happen? They will start releasing, or they'll start forming a thick wall. इसके चारों तरफ क्या हो जाएगा? Thick wall will be formed. A thick wall will be formed around the fertilized area. ठीक है. So there's amalgamation of contacts of two gamatangia, and finally they'll start secreting a common wall. And this is what is called as gametangial copulation. This is method is called as what is gametangial copulation. ठीक है. So this is seen in rhizopus and mucor. Mucor and rhizopus. So this is what. The next method is what? The fourth is spermatization. The fourth method is spermatization. मतलब यहाँ पे क्या होता है? जो male gametangia है, the male gametangia releases gametes. तो जो male gametangia है, it releases gametes. Releases gametes, which are non-motile. Okay, and we call them as what? We call them as spermatia. What do we call these male gametes as? They are called as spermatia. Okay, और यहाँ पे क्या है? फीमेल है ये देखो. This is the female. यहाँ पे हमारा एक्सेल है. ये हमारा spermatia है. Spermatia. Spermatia comes है. ठीक है, makes contact. ये देखो makes contact with the female gametangia and finally goes inside, fuses. And female gametangia is called as what? Female gametangia here is called as escogonia. This is called as escogonia. So spermatia will make contact with the escogonia and finally fertilization will take place and we get what is a zygote. And we get finally what is a zygote. So this is what is spermatization. And the last one that we have, the fifth one, what was that? Somatogamy. Somatogamy matlab there are no distinct male and female gametangia which are formed. Male and female gametangia are not formed. Not formed. यहाँ पे क्या होता है कि we do not have any well organized sexual organs. Yeah. There's no well organized somatogamy में there's no well organized reproductive organ which is seen. यहाँ पे क्या होता है कि somatic hyphae of two different strains. मैंने कहा ये हमारे hyphae हैं. एक hyphae ये आ रहा है यहाँ से. Somatic hyphae of two different strains will come together. ये negative strain का, ये positive strain का. So somatic hyphae of two different strains, the anastomose. The anastomose together, they come together. Okay, and common walls will dissolve. Nucleus of one will get transferred to another. Common wall is dissolved, and the nucleus of one, okay, one strain will get transferred into the other strain. So इसका nucleus यहाँ जाएगा, and now the dikaryoticization will take place. Dikaryoticization will take place, and finally followed by karyogamy. Finally followed by karyogamy. Okay, so मतलब fertilization will take place. Dikaryoticization, two different nuclei in the same cell, followed by what? Followed by nuclear fusion. So this is what is somatogamy. So we have seen different methods of sexual reproduction in the case of fungi. आपने एक चीज याद रखनी है, see there are certain terms. जैसे हमने देखा है lichens. Lichens are symbiotic structures. याद रखो. They are symbiotic associations between what? Between fungi and algae. याद रखो. एक बार क्वेश्चन पूछ देते हैं. Sometimes simple questions can puzzle you. So what is a lichen? It is a symbiotic association not between fungi and bryophytes. It is a symbiotic association between fungi and algae. This is what is a lichen. ठीक है. दूसरी चीज आपने एक टर्म हम यूज करते हैं नॉर्मली माइकोराइजा. Mycorrhiza. Mycorrhiza is what? Mycorrhiza is a symbiotic association between fungal hyphae and plant root. ये क्या है? This is a symbiotic association between what? Between fungal hyphae and plant root. This is what is a mycorrhiza. Very simple. आपका अगला लेक्चर कितने बजे है?
7:30.
अच्छा 7:30. चलो, I'll take 5-7 more minutes. We're about to finish. So this is what is a mycorrhiza. This is also a symbiotic association. अब आपने याद रखना है mycorrhiza. We know mycorrhiza can be of two types. याद. Normally mycorrhiza can be of two types. We have ectomycorrhiza and we have endomycorrhiza. Ectomycorrhiza and endomycorrhiza. Ectomycorrhiza में क्या होता है? जैसे मैंने कहा रूट है हमारी. तो ectomycorrhiza में what will happen कि fungi जो fungi है, it forms envelope. Fungi forms an envelope around the root. देख रहे हो? It forms a kind of envelope around the root tip. and it also penetrates. ये जो fungi है, this will penetrate into first few cortical layers. So it penetrates into what? Into first few cortical layers. This is very clear. ठीक है? And it will form a intercellular network. जैसे मैंने कहा ये हमारा cortex है, ये हमारे cortical, तो fungal hyphae penetrates into first few cortical cells and it forms a kind of a intercellular, मतलब between the cells. It will not penetrate inside the cells. So ectomycorrhiza में we have this sheath, this envelope of fungi. First few cortical layers are populated and it forms a intercellular. You देख रहे हो? This is a kind of an intercellular network. It does not penetrate inside the cell. और ये जो network है आपने याद रखना, this intercellular network, what do we call this as? ये question आ जाता है कई बार. This is called as Hutting's net. क्या नाम है? HAR. This is Hutting's net. एक term है Hutting's net. So what is a Hutting's net? It is nothing else. It is a type of ectomycorrhiza. ठीक है? अब देखो endomycorrhiza. Endomycorrhiza में क्या हो रहा है कि hyphae will penetrate the root hair, it will penetrate the epidermis, reach the cortex. It reaches the cortex also deeper. So reaches the cortex deeper and it penetrates inside the cell. मतलब सेल के अंदर भी ये जो hyphae, जैसे ये हमारा hyphae आ रहा है, fungal hyphae is here. It will penetrate inside the cell and forms knot-like structures. देखो, it will also form knot-like structures inside the cell. So starts penetrating inside the cell. So endomycorrhiza, we know it becomes intracellular. This basically becomes intracellular. So this is about the sexual reproduction that we have seen. We have seen the sexual reproduction, and now, before we wind it up, I have to tell you something. See, मैंने अभी आपको बताया था कि सोमेटोगैमी में जो लास्ट वाली सेक्सुअल रिप्रोडक्शन है, लास्ट सेक्सुअल रिप्रोडक्शन कौन सी थी हमारी? सोमेटोगैमी. Out of somatogamy, we get what? We get a dikaryotic cell. Dikaryoticization will take place and we get a dikaryotic cell with two different nuclei. We get a cell with two different nuclei. One is a negative strain, one is a positive strain, two different nuclei. Now this existence of two different nuclei in the same cell, क्या हो रहा है? Existence of two different nuclei in the same cell, this is called as what? This is called as heterokaryosis. This is called as what is heterokaryosis. So existence of two different nuclei within the same cell, we call it heterokaryosis. Is this clear? और आपने याद रखना है कि on the basis of the sex, fungi can be classified into three categories. On the basis of the sex, we can classify fungi into three different categories. The first one is what? The first one is hermaphrodite. Hermaphrodite, sometimes is also called as monoecious. This is called as monoecious. Okay, hermaphrodite. The second category is what? The second category is dioecious. Pehla monoecious or hermaphrodite, second is dioecious. The third category is sexually undifferentiated. What is the third category? Sexually undifferentiated. A category yogi, a category yogi, a category yogi. So on the basis of the sex, we can classify fungi into three types: monoecious, dioecious, and sexually undifferentiated. ठीक है? अगर हम बात करें monoecious और hermaphrodite की, मतलब यहाँ पे each thallus will have both male and female organs. Both male and female organs are present in same thallus. But whether they can fertilize each other or not, ठीक है? That is the question. Sometimes they can fertilize each other, sometimes they may not. So whether these male and female are compatible or not, it depends on the species. ठीक है? So this is what is monoecious. Dioecious में बोले तो dioecious में thallus will only bear one type of sex organ, only one type of. So thallus only has one type of sexual organ. It only has one type of sexual organ. Either it is male or it is female. Very clear. Okay, what is sexually undifferentiated? The name itself is very clear. कि यहाँ पे the sexually functional structures are produced. We have these structures, but we cannot morphologically distinguish them. याद रखो. We have these structures which are formed, sexual structures are formed, but they cannot be differentiated as male and female. We cannot say whether it is a male structure or whether it is a female structure. That is how we say sexually undifferentiated. Male and female cannot be distinguished, but we know they are different strains. So this is what is sexually undifferentiated. I'll 2 मिनट और लूंगा आपके. अब हमने देखा कि 3 तरीके की कंडीशंस होते हैं. 3 तरीके के सेक्स हैं, मोनोशियस, डायोशियस और सेक्सुअली अनडिफरेंशिएटेड. ठीक है. अब आपने याद रखना है कि दिस above 3 sex categories, above 3 sex categories that we have seen, these 3 sex categories, देख रहे हो? These 3 sex categories can again belong to 2 different groups. They can belong to 2 different groups. We can classify these 3 categories to 2 different groups. One is homothallic fungi. Homothallic fungi. One is heterothallic fungi. Very simple. When I say homothallic fungi, मतलब यहाँ पे the thallus is monoecious. Thallus can be monoecious, but it is self-fertile. मतलब thallus हमारा monoecious और self-fertile है. Homothallic आपस में ही इसमें fertilization हो गया. एक ही thallus में मेल है, मैंने कहा एक ही thallus है, यहीं पे मेल है, यहीं पे हमारा फीमेल है, and they fertilize among each other. So this is what is homothallic fungi. Heterothallic में क्या हो जाएगा? The thallus is sexually sterile. हम ऐसे कह सकते हैं thallus is sexually incompatible और thallus is sexually sterile in case of heterothallic fungi. मतलब यहाँ पे you will need 2 different structures. 2 different thalli are needed, one male and one female, क्योंकि thallus is sexually incompatible. So we need opposing mating partners over here. 2 partners are needed in heterothallic fungi. So am I clear with this? And finally, let me tell you something. What is parasexuality? कई बार क्वेश्चन आ जाते हैं parasexuality. ठीक है. Now, parasexuality is a process in which, so it is a process in which plasmogamy, karyogamy, and meiosis will take place. क्या मैं कह रहा हूं? It is a process in plasmogamy. मतलब fusion of 2 gametes. मतलब cytoplasm of 2 parental cell will come together. Plasmogamy, पता है ना? Plasmogamy, karyogamy. तो जब भी कोई रिप्रोडक्शन होता है तो क्या होता है वहां? रिप्रोडक्शन में पहले क्या होता है? प्लाज्मोगेमी. भाई एक सेल आया, ये एक सेल आया, इसका न्यूक्लियस है, इसका न्यूक्लियस है, क्या हो जाएगा? ये आपस में फ्यूज कर गए, साइटोप्लाज्म बिकम्स 1. इनके साइटोप्लाज्म बिकम्स 1, लेकिन न्यूक्लियस तो भी 2 है ना. साइटोप्लाज्म बिकम्स 1. सो दिस इज प्लाज्मोगेमी. दिस इज, कैन यू हियर मी?
Yes.
Just a minute. So plasmogamy takes place. ठीक है. Cytoplasm becomes 1 and finally followed by karyogamy. This is followed by karyogamy. So what we get? So this is a karyogamy. We get a 2N structure over here. पहले यहाँ पे N था, ये भी N था. Plasmogamy में karyogamy has taken place. We get a 2N structure, and now we know meiosis will take place. Meiosis takes place. This is what is parasexuality. मतलब plasmogamy, karyogamy, and meiosis, all the 3 will take place, but there is no fixed time in the life cycle. याद रखो, parasexuality में we have all the three processes which are part of sexuality, but there's no fixed time, there's no fixed juncture in the life cycle. So we call it what is parasexuality. Okay? So I'll bring this lecture to end over here, and we have nearly completed your fungi. I have to only talk about fungal classification. For that, I can give you a handout for fungal classification. That brings us to the end of the fungi. Okay? So I'll leave over here because a lot of students are coming in.
Thank you, sir.